So anyway, welcome everyone. It's nice to see your faces. Spring is in some areas arriving. And so of course with spring, the continual need for education continues. And so that's what we're doing today. I'm still in the US, and as I just said, those of you who heard, I just met up with Roxana in Orlando a couple of days ago. But where I am now in New Jersey, I'm just going to turn my screen around so you can see this pretty view I have from my hotel window with snow and sun. So, but I'm flying back to Copenhagen later this afternoon, and then from then on, hopefully I can say it's 4 o'clock Copenhagen time, but now it's 10 o'clock New Jersey time. So anyway, I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, a group of companies that is dedicated to change health and support optimal health through functional medicine, personalized medicine. And remember, practitioners, in your profiles, you can go in and check out the lab tests that we're going to cover today, and like any exciting supplements and so on. So make sure you have your logins so you can do further research in the VMS platform. Also, we will be recording, and it is recording already, and we will share the recording with all of you, and the slides will be shared as well. And then the topic for the next webinar, which is on the 7th of April, is on CoQ10. And you might think it's a little bit boring, just like CoQ10, but it's CoQ10 on the whole mitochondrial health. And the person who's going to talk about it has dedicated a lifetime in researching in CoQ10 and mitochondrial health, and was actually on the team in research where they got an award, so you will get a lot of knowledge, and I think this webinar will take you to a new level in regards to CoQ10. So anyway, the star of today's show is Dr. Julia Malkowski, who is a staff doctor at Doctors Data. It's a very good laboratory that we've been working with for many, many years. And they have launched the GI 360 test, and that's the one she's going to put in focus today. She has a doctorate in naturopathic medicine and in chiropractic medicine, so she's very well educated in our field, and her whole life is dedicated to living that way as well. Also her children, she pushes functional medicine thoughts and ideas onto them so that they can strive and be healthy. So, and just remember also to chat in the chat box, ask questions. I will answer if I can, or take notes of any interesting topics or questions that I cannot answer. And then after Dr. Malkowski's presentation, we will do Q&A as we normally do. So anyway, Julia, it's up to you to share your screen and take on the stage.
All right, thank you so much. Okay. Looks like there's a little bit of a technical glitch here. Hold on.
No stress.
Okay. Thank you for your patience. Okay.
Yeah.
All right, so can everybody see my screen?
It looks good, yes.
Okay, great. Thank you so much. All right, so thank you for that introduction. I'm so happy to be here today and speaking to you regarding the microbiome. This is going to be a great talk today. So our talk today is going to be about the microbiome, stress, and autoimmunity connection. Okay. All right. So today we're going to cover a few different topics, and we're going to try to put everything together. So the information we have here is going to be the dysbiotic gut bacteria, how they influence neurotransmitters and hypothalamic pituitary adrenal axis. So we really want to understand how everything is connected. We are going to talk about how stress does influence dysbiotic gut bacteria. We're also going to describe how stress and dysbiotic gut bacteria influences fecal secretory IgA, short chain fatty acids, which are very important for physiology. We're going to talk about the relationship here with intestinal permeability, inflammation, and then autoimmunity. We're also going to identify how gut bacteria can actually benefit stress-related adverse health outcomes and regarding further dysbiosis. And we're going to talk about the link between stress and pathogens, bacteria. And we're going to also talk about how intestinal permeability, again, is going to play a role here. So that's what we'll talk about today. So to begin, we are going to start with the wise adage, the father of medicine, Hippocrates, back in 3rd century BCE, he did state all disease begins in the gut. And we are learning now with the advent of technology and functional medicine, we're really learning how this is true. And so we're at this beautiful place now where we can really best serve our patients with the greatest technologies and understanding something that's held true since the beginning of medicine, that really the gut is that foundation. So let's get into this. So our topic today is going to be discussing the microbiota. The microbiota is a broad group of bacteria. We have microbiota actually all over our human body, from our skin to our upper GI to our mouth, everything. So our focus today is going to talk about the microorganisms within the colon. There will be viruses and archaea there as well, but we're not going to talk about those today. It's well known the microbiota within the colon does protect us from invasive pathogens. Additional considerations, though, are going to be the processing of bioactive nutrients, neurotransmitters, especially that's what we'll talk about today, but they're also involved in vitamins, and fatty acids and things like that. So there really are the cornerstone, the central beginning part of human physiology to a certain extent. Microbiota also influence neurotransmitters, histamine, gastrotransmitters. The microbiota actually communicate back to the liver and the stomach to secrete enzymes for digestion and things like that. So they're really playing a role all throughout the digestive process. Additional considerations include neuropeptides, steroids, endocannabinoids, and many more. One thing we will talk about today is how the microbiota are involved in priming the host immune system. And alterations here will trigger a wide range of physiological disorders. So considerations include low-grade inflammation, metabolic disorders, lipid metabolism, insulin sensitivity, and all of these are related to metabolic disease and autoimmunity. Okay. Specifically within the gut, we want to get to the core of what makes up the gut microbiota. The very first bacteria to colonize the gut is Clostridia. Clostridia is a broad group of bacteria and unfortunately well known for C. difficile, but that's actually just one of the Clostridia. There's hundreds. So the majority of Clostridia is healthy Clostridia, and that's really the first one we do see in the human colon in infancy. Two of the major phyla continued would be Bacteroides and Firmicutes. And then additional major phyla to consider here are the Actinobacteria, the Vermimicroba, the Tourniquets, and the Proteobacteria. So these are the main phyla that constitute the human colon. And we'll see here, these really do represent microbiome abundance and diversity, which is implicated in a variety of physiologic and health conditions, okay? Currently, there's more than 10,000 microbial species occupying the human ecosystem. It's about 2 to 4 pounds of bacteria actually in your gut. So that's a lot. Interestingly enough, the first 3 years of life are really actually crucial to lay the foundation for your adult microbiota. So really those first 3 years of life is when the microbiome is developing. And this is going to kind of lay the foundation for the rest of the life. And then here we see the development, immune system is developing, neurologic system are developing. So these are all developing together, and then the microbiota is there as well. So what we will learn about today is actually during that time period, we do want to consider actually stress. So laying the foundation for the HPA axis and the microbiome in relationship to that as well. So there's some interesting information about that. By the age of 2 to 5, really the human microbiome is the adult microbiome. So there's this window of period there that development. All right, so here we have a sample of the gut microbiome. This is organized by phyla. Now, this is important because when we're going to talk about the human microbiome and physiology, one of the primary considerations is going to be abundance and diversity. You would be hard pressed to find any study regarding the gut and human physiology that doesn't mention abundance and diversity. So healthy abundance, healthy diversity is associated with positive health outcomes. Decreased abundance and decreased diversity is associated with less beneficial health outcomes and pathogenesis. And so here we see the patient's result is that white web there, and it's superimposed on the normobiotic reference range, which is actually that green hexagon. So for this patient, we can see there's decreased abundance and decreased diversity. For this patient, the implications would be there's going to be some health implications. We'd want to get more information beyond this phyla, detailed information, but this is a great place to start. And just of note here, we do have some key bacteria within the phyla. So within the actinobacteria, we see the Bifidobacterium, and these actinobacteria are associated maybe more with vegetables and things like that. The Bacteroidetes, we do have those as well. Within the Firmicutes phylum are some very popular bacteria, including Clostridia, Lactobacillus, Faecalbacterium protenitzii, and Ruminococcus. So we'll get to learn more about these through some cases at the end. And then the bottom corner there, we see the Proteobacteria, which tend to house some of the bacteria associated with more inflammation. So the Escherichia and the Enterobacterales. So this patient, for this result, the first thing we know is decreased abundance, decreased diversity, and maybe some skewed toward inflammation. So we'd want to follow up and get more information and see what we can do here. So this is a basic introduction. So how exactly does the microbiome affect human physiology? Let's dive into that. The consideration here would be the microbiome gut-brain axis. So this is relatively new that we are discovering really the connection between the gut and the brain via the beautiful vagus nerve. And previously we thought that the brain talks to the gut and that's it. Everything all the directions come from the brain, but actually that's not true. So the gut actually communicates with the brain via multiple mechanisms. And that's the beautiful thing about functional medicine is really we can understand these concepts and really implement them to help our patients. Okay, so with the microbiome gut-brain axis, we're talking about the autonomic nervous system, the enteric nervous system, the vagus nerve, and of course the HPA axis. Prime consideration here is actually going to be diet, because diet is the environmental factor that actually links the complex crosstalk between the gut microbiota and the immune system. And that's really where we can help our patients is when we talk about the diet. So here we see a diagram, okay? The bottom portion here is going to represent the bacteria within the colon, and then the middle portion is going to represent the microbial mediators that actually travel via the vagus nerve to influence the central nervous system, to the brain right there, okay? So sometimes people will say, with regards to different functions within the colon, that those, serotonin within the colon doesn't actually travel to the brain. Right, but the microbial mediators do, and so they're signals. It's like a game of hot potato. They're signaling the information, and then that is how that impact is made. Okay, so of note here, we see the bacteria, and the most important thing here is going to be in the middle here, the short chain fatty acids. So the food we eat, the bacteria ferment this to create short chain fatty acids. Those short chain fatty acids signal to monocytes, dendritic cells, T regulatory cells, and in a healthy environment, when we're eating healthy foods, the retinoic acid is a vitamin A derivative. So in a healthy environment, these will signal and this will activate dendritic cells, monocytes, and this will actually decrease inflammation via TNF and nuclear factor kappa B, signaling to interleukin 10. And then that information will actually go to prime the T regulatory cells, the T17 cells. And so then you're going to prime a healthy immune system that actually is able to identify self and non-self. And so really this is where we are understanding how the microbiome does influence the development of the immune system, okay? So it's a beautiful, simplistic consort here. So now we understand about microbial host communication. Let's get a little bit deeper into the physiology here. So again, the important aspect here is going to be the food that we eat in a healthy environment. The bacteria ferment the soluble fiber to create butyrate. And then butyrate is the short chain fatty acid that will go ahead and feed the colonocytes. It will support this incredible barrier, and that healthy mucosal barrier is very important to prevent intestinal permeability and the pathogenesis of autoimmunity, which is what we're focusing on today. So here we see a healthy environment in which in butyrate abundant, we see the short chain fatty acid propionate there. All of those are in healthy values, and they're communicating to the different cells like the Paneth cell to create the mucus, and we have this healthy mucus barrier, and that keeps us healthy. You know, just of note here, there's so many different communications from the short chain fatty acids. Just one we have here is going to be creating those gut peptides that actually decrease our appetite and help with normal glycemia. So there's a lot of implications here. Of course, also too, like we said, that those lymphocytes are properly developed, and then we have a proper immune defense. Okay, so this is what we see in a healthy gut. This is what we're aiming for. So here's an example of what we would be considered dysbiosis induced breakdown. And unfortunately, this is probably very common. You might see this in your patients. So this is actually the development of intestinal permeability, inflammation, and autoimmunity. The first step in this inflammatory cascade is going to be the lack of soluble fiber. And one of the bacteria really implicated there is going to be the Akkermansia muciniphila. The Akkermansia muciniphila is a kind of rock star bacteria right now. We're learning more and more about how this bacteria really is responsible for contributing to that healthy mucosal barrier. So that mucosal barrier is very important here. When that mucosal barrier decreases, due to lack of soluble fiber and dysbiosis, especially implicated Akkermansia muciniphila, when that mucosal barrier decreases, then we see breakdown of really this whole system here. And so those cells are not properly fed with the short chain fatty acids, and then we do tend to see those tight junctions are going to break, and then we have intestinal permeability. And then this is even further worsened by some specific bacteria, inflammatory bacteria, Enterobacteria, et cetera. And then you have worse inflammatory cascades. So the implications here would be lack of regulation of those T cells and lymphocytes. So you'd see immune issues. You're not going to have those gut peptides, so your patient's not going to feel full after they eat. And then potentially autoimmunity as these bacteria enter systemic circulation. Okay. This dysbiosis induced breakdown is very important. It's, you know, it's very common. It's kind of where we as functional medicine providers can really help our patients, right? Once you have an understanding of this. So intestinal permeability is key when we're discussing the microbiome and autoimmunity. What contributes to intestinal permeability? Well, there's a lot of factors, right? So interestingly enough, that tight junction, that barrier, it's dynamic. It's not static. And so different things can change it. So alcohol, drugs, sugar, gluten, gliadin, the protein of gluten, those things actually do induce intestinal permeability. Okay, there is some evidence that glyphosate, so that can cause epithelial cell damage to that environmental toxin. certain cancer treatments and radiation. So that's just something to pay attention to. If your patient's undergoing those, you probably want to support this system, okay? And then of course, certain metal oxides, suffocants, sodium chloride, okay? So really, like we said, intestinal permeability is going to be that initiating factor for an inflammatory cascade. And then once we have intestinal barrier damage, we could see potentially further increasing intestinal permeability. So we're going to compound that. So we really want to address this as soon as we can. Of note, intestinal permeability is associated with actually advanced age. So maybe it's something to think about in your older populations. And then also type 2 diabetes, Crohn's, celiac, multiple sclerosis, IBS, and as we will see, actually stress. So stress will be an independent risk factor. Okay, so what do we want to do in our patients with intestinal permeability? What we want to do here is we want to actually take a minute and we want to talk about the diet. So like we said, the diet is going to be a factor contributing to intestinal permeability. So high fats diet here. So the photo shows actually a typical American diet, which is going to be like bread and a beer. Unfortunately, that diet in of itself will actually contribute to intestinal permeability. So in this case, we want to help our patients and we want to avoid alcohol, sugar, gluten, and things like that. So the considerations would be actually adding in soluble fiber. So we touched previously on soluble fiber, and that's going to be a theme actually throughout this talk because soluble fiber is very important to support a healthy microbiome. So soluble fiber, the considerations would be actually be 8 to 15 grams of soluble fiber per day. So it's a good amount of soluble fiber. Okay, all right. And then, so interestingly enough, some things associated with intestinal permeability would be extreme diets. And I know that those are popular now. There's a lot of people doing ketogenic diets, carnivore diets, intermittent fasting. So potentially, especially the ketogenic diet and the carnivore diet might contribute here, especially with regards to the lack of soluble fiber. Okay. All right. So another consideration with regards to intestinal permeability is when any bacteria adhere to the colonic wall. So remember previously we saw that the healthy mucosal barrier integrity, there is a nice viscous barrier there. And when that barrier decreases, if any bacteria adhere to the colonic wall, that irritation will actually induce intestinal permeability. And we're going to see that. We're going to see that in a minute. Okay. And then, of course, if your patient's diagnosed with a gastrointestinal inflammatory disease, autoimmune Crohn's, things like that, you know, that would be a consideration. Stress and oxidative stress as well. So you want to be checking your patients for oxidative stress. So here we have a beautiful example of intestinal permeability, and this is serum zonulin. So how many people are familiar with serum zonulin? Zonulin family protein is actually a byproduct of the intestinal barrier. When it breaks down, zonulin is a byproduct. And so we can actually measure zonulin to see how permeable the intestines are. So this is an excellent test. This is a direct test for intestinal permeability. It actually is going to give you the same results as if you performed the lactulose mannitol drink test. So it's really a clinical decision if you want to use the lactulose mannitol drink test or if you want to use the serum zonulin. Either way, you'll find out the exact same information. So this is a great test, a simple venipuncture, and we'll see there's quite a few patient populations. We did discuss previously Crohn's disease, diabetes, patients that are stressed, patients diagnosed with autoimmunity, things like that, those patients that have many, many food sensitivities. So there's quite a few implications actually for this novel testing. All right, so unfortunately, mucosal barrier and intestinal permeability is actually going to be common. I don't know about you if you've seen this, but I've definitely seen it increase. It seems like more and more people are actually going to be diagnosed with this. So we want to be able to help our patients. So what will we do? The first consideration, like we said, would be dietary changes. So we're going to move away from that standard American diet. We're going to avoid processed food, avoiding gluten and fats and sugars, things like that. Alcohol as well. You want to address the dysbiosis, so test and address, right? Remember, microbiome abundance and diversity is going to be the first consideration here, and then we're going to look at specific bacteria. We talked about that Akkermansia, and we will see this later in the case of ruminococcus. There's specific bacteria that contribute to mucosal barrier integrity. So in patients that we suspect intestinal permeability, that's going to be a component as well. So it's very appropriate to consider that. We want to consider supporting our patient digestively. Remember, the bacteria in the colon do contribute to digestion. So they do send signals to the liver and the gut and things like that. So if there's dysbiosis in the colon, you know, the whole digestive process can be affected. So we do want to support that, okay? We do want to decrease inflammation in the colon, because if there is shorter mucosal barrier integrity, then some bacteria could be touching the colonic wall and causing inflammation and irritation. Supplements here would be quercetin, vitamin D, and we'll talk about vitamin D, retinol, turmeric, gamma-linolenic acid, omega-3 fatty acids, so those EPA and the DHA, and of course, well-known aloe vera, okay? Additional considerations would be zinc, beta-carotene, pantenothenic acid, and now glutamine. So there's some studies there that those are helpful. Okay. So rest assured, there are things that can be done to restore the mucosal barrier. So that is the positive part here. Okay, additional consideration actually is going to be secretory IgA. So we do see secretory IgA decrease in these cases, all right? So during acute stress, actually fecal secretory IgA is upregulated. So that's interesting, but it does make sense because the body is brilliantly designed. So historically, our stresses were life-threatening survival type stress in the wilderness in terms of hunting and running and things like that. So during an acute period of stress, your body primes your system to fight off an infection. So if you were running from a predator or running during a hunt and you got cut, your body's primed and ready to defend itself. So during an acute period of stress, we see increasing fecal secretory IgA, and that is to protect us. Most likely, you're not going to see that in your patients. What you will see is this chronic stress. Chronic stress downregulates the immune response and downregulates fecal secretory IgA. So in these patients that are chronically stressed, they're going to have lower levels of fecal secretory IgA, and that's going to put them at risk for an infection. As we are going to see during a stressed environment, we want our patients their immune system to be the best as possible, because these brilliant bacteria are already ready for us to be stressed, because that increases their chance of survival. So in your patients that are stressed, you really do want to address fecal secretory IgA, right? Because that's one of the first priming immune systems right there, their first line of defense, okay? The additional considerations, HPA axis activation influences interleukins and secretory IgA production. So there is that connection there with HPA axis and secretory IgA production. So we do want to kind of look at these things together, I would say. And definitely secretory IgA is a component within that mucosal barrier integrity, and it does play a role in maintaining homeostasis. So in patients that have increased intestinal permeability, I would also consider fecal secretory IgA, okay? And again, secretory IgA does induce IgA and mucus secretion, and this promotes epithelial barrier integrity and prevents pathogen colonization. So there's a few different factors at play here, but we'll see they do all go together, because within the colon. It's an intricate system, but we do want to keep the IgA healthy. We want to keep that mucosal barrier, IgA adequate, that mucosal barrier healthy and adequate, and that's going to prevent pathogen colonization. In a patient that has low fecal secretory IgA, the implications would be lower mucosal barrier integrity, which and you have lower fecal secretory IgA, so that makes an advantage for the pathogen, for the bacteria, because there's no IgA to protect against them, and then they can contact that colonic wall and increase inflammation. Okay. So here we see a result. This is from a patient that reported stress, and I'm sure so many of your patients are reporting stress now during this time we're living, during this pandemic. The past 2 years have been actually very stressful for most people. So this patient reported stress, and we can see here the IgA is very low. Okay, so here's an example from a laboratory report. The first part is going to be the inflammatory markers: lactoferrin, lysozyme, calprotectin. So non-specific inflammation, but then we can get very specific, which with regards to IBS and IBD there. Now, so this patient doesn't have any of those diagnoses. They're just stressed, right? Like so many of us are. And we can see here the IgA is so low. So the implication here is, in the presence of this chronic lifestyle stress, the patient's going to be at risk for inflammation and intestinal permeability. The next consideration here we do note is the short chain fatty acids, right? And remember, from the beginning of this conversation, we discussed that the short chain fatty acids are just so important with regards to microbial mediators, but then they're also directly derived from the food that we eat. So we see here the butyrate is very low and the valerate is very low and the totals are low. So this patient's going to be actually at risk for inflammation because that butyrate is very low, and normally it would go and feed the colon and decrease inflammation and things like that. So this is a report that you will see in patients that are stressed. And the good thing is there's things you can do here to really help this patient to remedy this situation. So it's a good consideration. So what will we do for low IgA? Well, thankfully, there's a few different things we can do. The probiotics. So we're going to see more information about these probiotics, but the Lactobacillus rhamnosus GG and the Bifidobacterium lactis BB12 are very well known to have positive health implications. So those are going to be the bacteria that we're, the probiotics that we're going to use. Saccharomyces boulardii actually has been noted to increase secretory IgA, so that that yeast might be benefit there. And then of course we're going to add the prebiotics, so the soluble fiber, chickpeas, beans, red kidney beans, things like that. So inulin would be the supplement that people are using generally. So we want to get up to 10 grams a day. Most people are not there. So if we think about it, I'm sure you've asked your patients about their diets and how much fiber they're eating, and you'd find out people are having, total about 10 grams of fiber a day. Generally speaking, fiber for fruits and vegetables, it's going to be 1/3 soluble to 2/3 insoluble. So if people are getting that 30 grams of fiber per day, about 10 would be soluble. So that makes sense, actually. But now we really do actually need to probably talk to our patients about making sure they get that 10 grams of soluble fiber per day. So important. Additional considerations include glutamine. Here we see again omega-3 fatty acids, olive oil, zinc, vitamin A, and D3. So those will help with intestinal permeability as well. And then of course, like we said, chronic stress and low DHEA decreases secretory IgA. So another indication here for testing cortisol and DHEA. So everything goes together. All right, so stress. Unfortunately, like we touched upon earlier, we're not living in life-threatening stress in terms of survival in the wilderness, but our modern life induces a low level of stress on an everyday basis. This is stress our body's actually not meant to deal with. So we're meant to deal with stress in acute situations, and then we compensate. But right now, especially within the past few years, everybody has just been stressed almost every single day. And I'm sure you've seen this in your practice when you ask your patients to rate their stress on a scale of 1 to 10, right? People are very stressed, okay? And so that's characterized by increased sympathetic nervous system and decreased parasympathetics. In these situations, people aren't going to be able to digest their food. They're in fight and flight. They're not in rest and digest. We're going to activate the hypothalamic pituitary adrenal axis. And then we'll see decreased fecal secretory IgA, decreased GI microbial composition, decreased short-chain fatty acids, and increased intestinal permeability. So these, specific GI conditions are associated actually with stress. So definitely a key link here. Here's a beautiful study that was recently done on regarding stress and intestinal permeability. So they measured the Norwegian army that was training in the Arctic, and they measured their microbial composition, and they noted that low microbiome abundance and diversity in the presence of stress increases intestinal permeability. And then the participants reported GI issues, loose stools, abdominal upset, things like that, which I'm sure you see in your practice. And so really the consideration here, the conclusion was that in a stressful environment, right, they're training, they're in the army, they're in the Arctic, it's not very hospitable. In a stressful environment, you do see increased intestinal permeability and GI distress, okay? So really the considerations here is decrease abundance and diversity, like we talked about previously, and then we see intestinal permeability, and then in this case, the patient's presented with GI symptoms. So this is a good study just to show exactly, stress does increase intestinal permeability, decreases abundance and diversity within the colon. All right, so here we have a photo here about, this is kind of like how we think about intestinal permeability. What should stay in that bucket there should stay in the colon, but it's leaking out, right? Okay. All right, so now we're going to talk a little bit about when we are stressed, how does it change our gut microbiota? All right, so we know that almost half of the norepinephrine is actually made within the gut. So the gut is actually a factory for dopamine, serotonin, right? So it is important here. Now, these bacteria within the colon themselves, they can actually secrete their own catecholamines, and those bacteria are Bacillus, Proteus, and Serrata, and Staphylococcus aureus. These bacteria actually synthesize their own catecholamines that we as humans, our body can't tell if this is exogenous from the bacteria or our own endogenous. So the bacteria do play a role in there. Now, butyrate is really important here because butyrate in the presence of Clostridia is going to enhance the transcription of tyrosine, and then that's associated with serotonin synthesis. So really the gut microbiome, they really play a role here. All right. Now, so when people are stressed, you might not know this, but when people are stressed, what happens is those catecholamines actually spill from the systemic circulation into the colon. The bacteria love this because in the presence of these catecholamines, they can actually communicate to one another to enhance their survival. So of note, the salmonella will actually transfer a plasmid to E. coli to help it to increase its survival. So it's pretty interesting. Now, during these situations of stress too, the commensal bacteria will express beta-glucuronidase, and then beta-glucuronidase will go in and cleave the norepinephrine and dopamine from that were conjugated, and now they're free floating within the colon. So in the presence of stress, you have a variety of different mechanisms actually increasing these catecholamines within circulation. All right. So another consideration is, generally speaking, the bacteria, the lactoferrin and the transferrin, they limit iron, so it makes a bacteriostatic environment within the colon, so that way the body is protected from infections, from pathogens. Unfortunately, during the time of stress, like we said, the catecholamines are within the colon. In the presence of these catecholamines, the pathogenic bacteria they see these catecholamines and they actually release factors that actually interact with the lactoferrin and the transferrin. And so instead of being bacteriostatic, now these pathogenic bacteria will actually they feed on the iron as a source of energy. So in the presence of stress, these pathogenic bacteria are able to create an energy source in an environment that would normally be inhospitable to them. Additionally, in the presence of these catecholamines, the bacteria enhance virulence factors, which help them to attach along the colonic wall. So the bacteria can attach along the colonic wall, and then that's going to lead to inflammation and intestinal permeability, right? Now, studies have shown that even several hours after a stressful event, the catecholamines and this pathogenesis is still occurring. So you might have driven to work and had a stressful incident like a road rage or something like that, and you're sitting at work, but you didn't even know that many hours later your body's still reacting, right? So at 4 hours after this, it's noted Escherichia coli is stimulated to grow. So the environment is great at that point. And then also a conditional consideration is that the environment actually enhances the Shiga toxin expression. All right. And like we said, the catecholamines, they do at this point, they're able to attach to the gut mucosa. This is going to lead to intestinal permeability, that inflammation there. And then the consideration is when you have those bacteria within the colon, they attach to the colonic wall, we have intestinal permeability. Then the consideration is these bacteria have opportunity to enter lymphatic circulation and systemic circulation, right? And that's going to maybe lead to autoimmunity. Okay. And then they're going to actually, during the process, like create lesions and destroy the gut tissue as well. So that's going to cause further problems. All right. So what are we going to do here? Let's talk about the microbiome, because this could be a consideration to actually really help the situation we have here. So with regards to the microbiome, we want to talk about HPA access. We want to talk about neurotransmitter dysfunction. We want to talk about the sympathetic nervous system, microbiome abundance and diversity. So we're going to definitely talk about that. Some key bacteria, lactobacilli, Clostridia, Bifido. Like we said, intestinal permeability, fecal short chain fatty acids, psychobiotics. We'll talk about those. And so really the question I want to present today, in patients with stress, is adrenal support sufficient, or should we consider stool testing? Okay. Here's just a note here about vitamin D. We did talk about it briefly, but vitamin D actually is going to play a role with autoimmunity and immune function. And the interesting note here is that actually vitamin D plays a role with intestinal permeability and also the development of immune function, and the gut is actually a cornerstone within that. So microbiome abundance and diversity is associated with proper vitamin D status, which is associated with proper immune function and autoimmunity. So another implication here for vitamin D to help in this manner. Okay. So enter into psychobiotics. Psychobiotics are a new term for bacteria. These are bacteria that infer a mental health effect, and people that are studying these are now thinking we should actually consider prebiotics as well into this. So the bacteria and then the prebiotics. Of note within this realm, we do talk about the lactobacillus. So the lactobacillus is a key player here. It's been noted to lower corticosterone. In studies, in animal models, lactobacillus and bifidobacterium were able to lower anxiety scores for 2 weeks. So that's a good period of time. They decreased anxiety and depression-like behaviors as well, Lactobacillus invictus and rhamnus, and potentially a neuroprotective associated with tryptophan and serotonin synthesis, okay? In one study, they had children consuming prebiotics. So again, prebiotics can fall under this category of psychobiotics. This noted a significant decrease in plasma interleukin-6 and a significant increase in Bifidobacterium. So we do want to increase Bifidobacterium, and we want to decrease that interleukin-6 inflammatory bacteria. So Bifidobacterium has shown of benefit. Bifidobacterium infantis actually was noted to improve immune system abnormalities and depressive-like behaviors in adulthood that was a result of early maternal separation in a murine model. So remember we said earlier that in the beginning of life is when your immune system is developed and your microbiota is developed. But here there are some studies that show that actually stress in early development can be, the physiological effects may potentially be remedied by psychobiotics later in life. So it is interesting to see a role here for the microbiota in this way. And although the next point here tells us that when you use these Bifidobacterium early in life, the effect is greater. So there might be a consideration here to really use psychobiotics early in life. We'll see, obviously more research is needed, but this is where we're at. Yeah, and so proper colonization early in life will serve to influence the development of proper HPA access. Just of note here, consider method of delivery, right? So C-section, we know that C-section births are very different, microbiome development and vaginal birth. So another consideration. Again, the major key players here are going to be Bifidobacterium and lactobacillus. And we see these, kind of these keep popping up. And again, we said that when this is a monkey model here. And so when the monkeys were separated from their mothers in early childhood, they displayed anxiety and stress and things like that. But then the Bifidobacterium and the lactobacillus were able to improve that, okay? Of note here that when in the monkey model, when they were stressed early in childhood, that the lactobacillus actually made decrease into adulthood. So there is a developmental stage there. And then in these studies, we also noted intestinal dysbiosis, low-grade inflammation, and so consideration again would be intestinal permeability. All right. And so we do know that with regards to our patients, right? So we do know that patients that have been diagnosed with depression and IBS, we do note that there's a connection there with gastrointestinal dysbiosis, chronic low-grade inflammation. So we want to consider this. And we do know that lactobacillus has been shown to lower corticosterone. So that would be a consideration here. Again, supplementation of Lactobacillus helvictis and Bifidobacterium longum decrease those test scores. So there's great promise here for these as psychobiotics, okay? The helvictis, lactobacillus, and bifido. All right, so here we see a test result, and this is just an example of a patient that was very stressed. And this patient, she reported great marital strife. Unbeknownst to her husband was advertising himself on dating websites, and her primary care doctor was able to help her because her chief complaint was GI issue, right? And you can see here, those bacteria, the Bifidobacterium and the lactobacillus, those are low, right, on this culture report, whereas those healthy, we see healthy Bacteroides, healthy Escherichia coli, and healthy Clostridium. So addressing this is really going to help that patient because they're having low beneficial bacteria here, and specifically in these bacteria that we do know are influenced by stress. So we want to help our patients in every way we can. Another consideration here is going to be inflammation. Unfortunately, with the standard American diet, we have low fiber, high fats, high proteins, and this is potentially associated with inflammation. I talked briefly about these fad diets, carnivore, keto, things like that. So these are just going to all be things that we want to consider as patients, and we really want to decrease inflammation because inflammation, again, is associated with metabolic endotoxemia, and in an inflammatory environment, we want to consider intestinal permeability and things like that. Okay, so now we have a chance to look at some interesting cases. Right. So here we have a 30-year-old male reporting stress. This is a patient that has the general stress that most people are dealing with now: lifestyle, occupation, pandemic. Remember, the first piece of information we have here is abundance and diversity. So for this patient, the web is going to be the white surface area is the web, and it's superimposed on that reference range, which is the green hexagon showing healthy abundance and diversity. The great thing about this report here is that it is really dynamic and individualized. Every single report is specific to your patient. So this is really individualized medicine here. So we can see exactly for this patient, we have decreased diversity. Abundance looks good because that white web is a large amount of surface area, but we see decreased abundance. The score is 1, the score is 3. So the beautiful thing about GI 360 is all of the bacteria that are associated with a healthy normobiotic gut or a dysbiotic gut, the information is calculated into a dysbiosis index score for your patients. Those scores go from 1 to 5. 1 and 2 are healthy normobiotic guts, and then 3, 4, and 5 are dysbiotic. So for this patient here, we see the score is 3, and that's going to be dysbiotic. So let's see. All right, so the key findings, so like I said, abundance and diversity is where we begin, but then we can get deeper into it with regards to specific bacteria. So recall, this is a 30-year-old man. He's really just stressed, just regular life stress. And we're going to take a look here. So we see here the bacteria, the Allostypes is low, Firmicutes is low, Bacilli class is low, but then we do see the Faecalibacterium protenitzii and the Ruminococcus navis. So those species are not at optimal levels, and that is actually associated, those 2 species, with mucosal barrier integrity. So recall, decreasing mucosal barrier integrity potentially leads to intestinal permeability. So we have a patient here that's stressed, and we see key bacteria associated with mucosal barrier integrity are out of balance for this patient. The next piece of information we have is we do see carbohydrates. So carbohydrates should not be in the colon. Now, maybe this is a carbohydrate malabsorption issue, but potentially this is actually a signal for us regarding intestinal permeability, because if there's intestinal permeability in the small intestine, then there's lack of surface area for those carbohydrates to be absorbed, and we will find them in the colon. So here's a clue here for this stressed patient that we do want to consider intestinal permeability. We also see this lower valerate. So in that case, we do want to, in this case, we do want to think about the short chain fatty acids and things like that. Okay, so here we have a look at the Firmicutes phyla. So for this stressed patient, remember we said the Faecalibacterium protenitzii, that's the lowest. It's very anti-inflammatory. It does help with mucosal barrier integrity. So this being low, the patient is going to be at risk for inflammation, decreasing mucosal barrier integrity, increased intestinal permeability. The patient's also eating more towards the standard American diet. So we do see this Eubacterium hali here is lower when people eat excess proteins. So that's something to pay attention to. The lower Clostridia denotes low soluble fiber. And then the higher Ruminococcus at the bottom denotes issues with integrity of the mucosal barrier. So this bacteria could actually just be eating that mucosal barrier. So this looks like a patient that we do want to say, okay, let's address your stress, but let's also address your microbiome. So, excuse me, my computer is running low. I'm just going to have to grab my thing. Sorry about that. Thank you. So for this patient, you do want to add the soluble fiber, like we said, just because that's going to be key to everything here. And we do want to address the mucosal barrier integrity, like we discussed. And in this patient, you probably want to actually run a zonulin test or a lactulose mannitol drink test. So that would really be the next steps here. Okay, thank you. Oh yeah, and like we said, the patient does have elevated carbohydrates. And so that's an indication of mucosal barrier integrity issues and intestinal permeability. All right. And then the next case we have here is a 46-year-old female. Her diagnosis is IBS. So this is a patient that's been diagnosed with IBS, and she's in the care of her functional medicine provider. So the first piece of information we have here is the web. The web is actually representation of abundance and diversity, and we see its diversity is disrupted and it's skewed toward these proteobacteria here, and those are associated with inflammation. The score is 5, so very dysbiotic. All right. So for this patient that's seen her functional medicine provider for IBS, it's a great thing she actually did the GI 360 because we'll see there's quite a few findings on here. The first piece of information we have is going to be the proteobacteria. These are very high. These are associated with inflammation. And then the Clostridia class is low. So we do want to help support that bacteria with soluble fiber. Streptococcus is very high, so that is high as well. So really, this looks like a slight inflammation profile that we want to add soluble fiber. But then when we look over here for the patient, we do note the patient actually has Salmonella and Giardia, and then we have 2 dysbiotic bacteria, Citrobacter and Klebsiella. So this is a great example of utilizing GI 360 because it has a test for pathogens as well as looking at the bacteria and their values, but we also identify pathogens because you do need to note that the patient has Salmonella and Giardia, and you do want to treat that as appropriate. Additional considerations include the Citrobacter and the Klebsiella. So those cultured bacteria should be treated as well. And then there's a yeast there. Lactoferrin is elevated, which actually does make sense in the presence of these pathogens. And here we see one positive thing, secretory IgA is elevated. And that actually does make sense for this patient because in the presence of the pathogens, we do want to see an elevated secretory IgA. Recall that secretory IgA is immune function, and so we do want to see that elevated. So that's great here. And here we see the GI, the pathogens panel portion of the GI 360. And like I said, the patient has that Salmonella and the Giardia. So we do want to consider the history for this patient and address these accordingly. All right, so putting it all together here, the important considerations are that dysbiotic bacteria does influence neurotransmitters, HPA axis, fecal secretory IgA, short-chain fatty acids, intestinal permeability, inflammation, disease, and autoimmunity. The gut bacteria and the catecholamines interact regularly. So we do want to think about the gut microbiome with regards to stress and adverse health events, considering dysbiosis and increased intestinal permeability. So we want to think of these systems all together. Intestinal permeability may contribute to autoimmunity. So in patients with either, in patients with autoimmunity, you want to consider intestinal permeability. With patients with intestinal permeability, you want to consider autoimmunity. And the gut catecholamines, stress, intestinal permeability, these are all closely interconnected and should be addressed as such. So we want to consider stool testing in patients with reports of stress. I would definitely consider that for sure. And then also with autoimmune conditions. All right, and now we do have time for questions. Thank you.
Excellent. Thank you very much for a very, very good presentation. And so we've got some questions that comes up at the chat, and there seem to be some confusion about Akkermansia, because every time we read an article or we hear a webinar, it's always focused on low Akkermansia. But actually, a lot of the practitioners here see high Akkermansia. Would you mind commenting on this, and what does high mean? How do we maybe treat it? How do we consider it and evaluate it?
Yeah, that's actually a great point. And so actually studies have shown that when Akkermansia is high, it will actually eat mucosal barrier integrity as a fuel source. So when it's too high, the consideration would be low mucosal barrier integrity. So when it's too high, it's not recommended to eradicate it, but it's recommended to look at the other bacteria and address accordingly. I think in that case, we saw Akkermansia was high, but there was also Clostridia was low. So we want to balance it out. And then also I would consider supporting mucosal barrier integrity as needed at that point. Yeah, good question.
And then, so actually in regards to this, because the question is, how do we do this? Like what supplements, what dietary, lifestyle, and so on. And for those of you who hasn't seen the webinar that we did some weeks ago from that we did on different supplements, it's called Healing the Leaky Gut. You can maybe get some inspiration there. So basically do the test, understand what goes on in the gut, and then use some of the clinical input that we have in some of the other webinars. So then a question comes up about Bifidobacterium biforme, what that is, what does it do? It's because Ingrida has a patient who has this high in culture. Are you able to comment on that, or do you need some support from
Yeah. I'll have to look. Bifidobacterium biforme, is that the one?
Yeah.
Yeah, I'll have to check. I believe that's a commensal bacteria, so I don't believe it would be something that you would eradicate. Yeah, but I'd have to double check on that one.
Mm-hmm. So then one is asking if the catecholamines show up in the test result, but it doesn't. But so I guess you would have to just consider, is this patient stressed? What does the test result look like? If this test result is skewed, you must assume that there is some form of stress response going on, or.
Yeah, that's a great question. It would probably be wise to do the, the HPA axis kind of adrenal function testing, and things like that. Maybe do neurotransmitter testing in conjunction with. But yeah, we're not going to measure them in the colon, and yeah.
Okay, super. So just a reminder for where you find the Healing the Leaky Gut. If you log into your VMS account, then you find education in the menu, and you log in there, and you can see it. You can see all the webinars that we have had throughout the last couple of years are there as a great resource. So that's the best place to find it, I would say. The YouTube channel is also open, but it's easier just to go in. They're all in there. It's easy to look around there. So the difference between the GI 360 Essentials times 1, 2, 3 and GI 360 Microbiome and GI 360 1, 2, 3. Can you quickly recap that for all of us?
Yeah, great question. So really the GI 360 is the test to start with. That test includes everything. So the first part is that microbiome abundance and diversity, that web and that score. So don't discount that, because it's very simplistic presented, but it's actually really advanced information. And like I said, abundance and diversity is really the cornerstone of the gut. So that's the first information within the GI 360. And then you'll have, like I did show the key findings, and then the bacteria that contribute to the score will all be listed. And so that's where you'll get kind of the meat and potatoes. But the GI 360 includes everything. So it's going to be microbiome abundance and diversity with the specific bacteria. It's going to include the pathogens panel, the parasites. It's also going to include culture for dysbiotic bacteria, commensal bacteria, yeast. Any dysbiotic bacteria or yeast will have sensitivities. And then the stool chemistries portion. So IgA, digestion absorption, short chain fatty acids, and beta glucuronidase. Now, the other profiles, the essentials and the microbiome, are really made for reordering. So you don't have to order the whole thing next time if you don't want to. You have choices. So the GI 360, yeah, so
I've been cheating here because I just looked it up so that you can see what it looks like in VMS.
Yeah, that's helpful. Thank you.
So the GI Essentials is the with 1, 2, and 3. That depends on how many times you want to collect for parasites.
Correct.
So the numbers 1, 2, or 3 is actually the days of collection. So if you choose 3, the patient needs to collect for 3 days, because they need to check out for parasites 3 days, but you can choose to do it just 1 day. And that goes for the GI 360 1, 2, and 3 and it goes at, now I have the Danish prices, so you can kind of like look at the prices as well. So the GI 360 is the most expensive one. That's the one that contains most information. Whether you choose 1, 2, or 3 collections, the price is the same. So so the essentials is bacteria and parasites and yeasts.
So not the stool chemistries.
But not the stool chemistry. So where you both do culture and you have the microbiome there as well. And then the microbiome is only the microbiome evaluation. And then the GI 360 is the microbiome evaluation and the culture and the parasites and everything else. But you need to, is the zonulin included?
No, so fecal zonulin is an add-on for all of the tests.
Yeah. So, oh yeah, here it is. So you can add on H. pylori and you can add on GI 360, oh sorry, zonulin to it. And then a question opens up, because then you mentioned serum zonulin, and then, but it would be easier to add stool zonulin, but when should we?
Yeah. So it really depends on the collection method. So if you want to add it on the stool test, that's fine. The stool, like I said, it's a dynamic system, so it does fluctuate, right? So if you're going to collect and your patient had a burger and a beer the night before, that permeability might be there. So it's a dynamic system, it changes. So I would just consider that, when you're using the stool one, but for ease of collection, it's a great option. Yeah.
Okay. So then a question came up. What does an ideal web look like?
Yeah, so it's actually just that green hexagon there. So it's going to be a hexagon, meaning that everything is essentially equal, and then it's going to be you'll see the green hexagon there. So it's right at that green hexagon. Now, I will tell you, I don't think I've seen any. I've seen some close, just because most people are pretty unhealthy, and the patients that come to us are the less healthy patients, right? So so it's that green line there. So in this report, right?
If it's in this area.
Well, it's actually perfect. Yeah, it would be perfect if so the Firmicutes is good. It looks like the Proteo is okay. Tourniquetes, I mean, that's kind of making a hexagon at the bottom there, but then we see lower actino, higher vermimicroba, lower bacteroides. So we don't see a hexagon, but this is, it's close, but it's not a hexagon. Sometimes you'll see that shape will make a triangle, it'll make a square, it'll make a rectangle. Sometimes you'll see that shape is really teeny tiny pulled in. So it's, again, specific individualized medicine for your patient.
Super. Then let me go back. Another question came up in a patient that was positive for salmonella but doesn't want antibiotics. How would you, because it's also a question of how sick and ill is the patient, what's the symptoms?
Yeah, exactly. So you want to consider the presentation and the history. And then also with that pathogens panel, everything is so sensitive and specific. So you want to, again, consider history and presentation for the patient. With regards to treating beyond the standard of care, I wouldn't really be able to elaborate on that. But I would say definitely consider the patient's history, presentation, and then also you want to look at IgA, right? So the good thing was in that patient, they had an IgA response, which makes you think that they are actually fighting the infection. So yeah.
Super. I think it's time to finish. I think I've answered lots of, taking up some of the notes that came up in the Q&A and in the chat when you were talking. So I think we've learned a lot. We've been through a lot. So thank you very much. And thank you everyone for staying on the webinar and learning. It's always a great learning curve. And it's nice to see the way that you come from like the lab point of view. And then we have information like what's the treatment, how do we do this? But the interpretation of a test is very important. And then we can move on to the treatments and get information other places. So, but thank you very much. And hopefully I will be able to invite you another time for another webinar on another topic. We will see, but probably still stool related.
Yeah,
so
absolutely.
Thank you everyone for staying on, and thank you, Julia, for your time.
Thank you, Anne-Catherine, and thank you to everyone today. I appreciate the attendance.
Bye, and thank you for being live with your videos. I like that.
Yes, very good.
Bye.